Ovary. Ovum mature in a follicle lined by cells granulosa, in which produce estrogen. After ovulation, the remains of the follicle becomes the corpus luteum, secretes progesterone for 12 days following ovulation. Follicle corpus luteum secrete inhibin. Functions of estrogen and progesterone. Development of female reproductive system and physical, including adolescent bone growth. Estrogen regulates menstrual cycles, sustained pregnancy, prepares mammary glands for lactation. Inhibin suppresses FSH secretions from anterior pituitary. Testes. Microscopic seminiferous tubules produce sperm. Tubular walls contain testicular cells. Leydig cells, interstitial cells produce testosterone. Lie in clusters between tubules. Testicular hormones. Testosterone and other steroids from interstitial cells nestled between the tubules. Stimulates development of male reproductive systems in fetus and adolescence and sex drive. Sustains sperm production. Inhibin from testicular cells limits FSH secretion in order to regulate sperm production. Skin. Precursor for vitamin D to absorb calcium. Keratinocytes convert a cholesterol-like steroid into cholesterol using UV from sun. Liver, involved in the production of at least 5 hormones, converts cholecalciferol into calcidol. Secretes angiotensinogen, a pheromone. Precursor of angiotensin II, a regulator of blood pressure. Secretes 15% of erythropoietin. Stimulates bone marrow. Hepcidin promotes intestinal absorption of iron. Sorts of IGF that controls action of growth hormone. Kidneys plays roles in production of 3 hormones. Convert calcidol to calcitrol. The active form of vitamin D increases calcium absorption by intestine, inhibits loss of urine, secretes renin. That converts angiotensinogen to angiotensinogen 1, angiotensinogen 2 created by converted enzyme in lungs, constricts blood vessels and raises blood pressure, produces 85% of erythropoietin, stimulates bone marrow to produce RBCs. The heart. Cardiac muscle secretes atrial natriuretic peptides in response to an increase in blood pressure. These decrease blood volume and blood pressure by increasing sodium and water output by kidneys and oppose action of angiotensin II. Hormone chemistry. 3 chemical classes: steroids, lipids, derived from cholesterol, secreted by gonads and adrenal glands. Peptides and glycoproteins, along with monoamines, are non-lipids, so they're proteins. Peptides and glycoproteins created by chains of amino acids, water-soluble, secreted by pituitary and hypothalamus. Monoamines derived from amino acids, water-soluble, secreted by adrenal, pineal, and thyroid glands. Hormone transport. Most monoamines and peptides are hydrophilic, which they like water, mix easily with blood plasma, water-soluble, travel in plasma as free hormones. Steroids and thyroid hormone are hydrophobic, doesn't like water, binds to transport proteins. Bound hormones have longer half-life, protect from liver enzymes and kidney filtration. Hormone receptors. Hormones stimulate only those that have receptors for them. Receptors are protein or glycoprotein molecules on plasma membranes in the cytoplasm or in the nucleus. Usually, each target has a few thousand receptors for a given hormone. Receptors act like switches during metabolic pathways when hormones bind them. Receptor hormones exhibit specificity and saturation. Proteins are specific equals receptor are specific. Saturated when all receptor molecules are occupied by hormone molecules. Intracellular receptors and direct gene activation hydrophobic hormones. This is the slower process of the two. Steroid hormones and thyroid hormones penetrate plasma membrane and attach to the intracellular receptor, enters nucleus, acts directly on the genes changing target cell physiology. Estrogen, progesterone, thyroid hormone act on nuclear receptors to take several hours to days to show effect due to the lag in protein synthesis. And then second messenger systems. It's faster. Peptide hormones bind to the cell surface receptor linked to the second messenger system on the other side of the membrane, activates G proteins, activates adenylate, silate, produces CAMP, activates or inhibits enzymes, metabolic reactions, synthesis, secretion, changes in membrane potentials.